Andreas, welcome to the Collaborative Research Center, Origin and Function of Meta-Organisms. It's a great pleasure. You are one of the leading host-microbe symbiont biotechnologists around, and you just gave us, in a seminar series, you gave us a wonderful, exciting, fascinating, multi-facet talk on what you have learned in the last decade on symbiotic bacteria, what they can do so that insects can eat, live, invade habitats and food which they, without the symbionts, never can do. And of course, insects are meta-organisms as all other organisms as well. And so there's many joint interests in that field. And it's very nice. And I'm super happy that we can talk a little bit about yourself, your research, and maybe also some more visionary questions, and also addressing maybe the situation of the junior generation. Usually, it's always interesting to ask and to find out, how did this person who presents such a wonderful broad spectrum of scientific ideas, results, papers, perspectives, how did it get to there? What does it need? What was it in your case, Andreas, which made you getting fascinated by what you're doing right now and by the index? Was there anything specific, or how did you move into that?
So I started when I was a child to collect butterflies and insects. So I think I started with 10, but first in empty cigar boxes from cigars. Then with 12, I had the first really glass boxes. I started to collect, and this is fascinating because I had also a book. So I have a diary, so everything what we have collected. So I can document what kind of butterflies I found at this time at certain places. And now, so many years later, when you look at the same, go to the same places, you would never get these insects, really, this diversity. So I can easily, from my own experiences, say we have an insect decline. So, and then this, I was also interested in fish, as you know. So I started to breed fish and make quick. So this was this was very early that I decided also to become a professor and to teach in zoology. So this was I can document this with that I really was that was a plan as long as I can think.
So yeah, fascinated by nature, and then somehow you got attracted specifically by the insects, and so then we can talk about your research in a minute. But let me say that your personality and what you are now here presenting as a scientist, which uncovers so many unknown details of animals. It is, as you have said in your lecture, it is only possible because you know the animals so well. And I cite now your lecture. You said there is not only a decline in insects, but there is also a decline in entomologists. And so if you think back, and also as now you are a university teacher, isn't there also a message for the younger generation that it needs more than understanding nucleotides and cell differentiation? It needs an intrinsic interest in the biology of an organism to really, at the end, exploit that organism for the better kind and for human.
Yes, I think that I would like to attract, especially students, to let's say to learn the diversity of insects and to become at least a part expert in taxonomy. I think, to my opinion, the entire taxonomy and systematics, the knowledge that is, let's say, written in so many books, it's a culture. that we lose if nobody is able to work with this one. We are really losing expertises, and because nowhere, I was in Potsdam as a professor for zoology and evolutionary biology. I was the last one who taught all of these courses, let's say the field excursions, taxonomic, everywhere. Berlin, look around, so these courses are missing. And I started here in Gießen again to re-establish and to expand taxonomic courses, even for the agricultural students and for the biologists. And I got a very good feedback for this, as I said. And we are inspiring also others. For example, Baden-Württemberg, Herr Kretschmann, has also provided 2 professorships now for the University of Hohenheim, taxonomists. They're also able. So we inspire also others that you have to go in another direction. We are thinking about to apply for a graduate school together with Senckenberg, where we combine all the experts that we have. That's the reason why the collaboration with Senckenberg is so exciting, and why Senckenberg has, let's say, funding a professorship for it, because they were really saying that there is no other university doing taxonomy, and we try to promote this. And that was one of the reasons why Volker Moosbrugger came to this idea. Okay, then we will go and for your new institute to establish this and to expand this. And we are planning a graduate school to train and to attract offspring, that in the near future you have experts. You know by yourself, by your experience, where are the experts that can go out in the field and determine any bug on everything that you can see? There's missing. And with the lack of this knowledge, this is almost all universities I know, it's the same story. And now we, due to the insect climb, there's an interest. And also the industry has interest now to have, because when they, for example, a big company is developing insecticides, and you have a longer, the risk assessment involves also studies where you need experts, and there's luck. It becomes more and more difficult. So we got a contract to explore the metagenomics and meta-barcoding as an alternative. It's a cheap and more efficient to compensate for the lack of experts. That is unbelievable.
I think this is a very important point, Andreas. My colleague and friend Margaret McFonay in Hawaii, she talks and she promotes, she calls it the teaching of a new biology. But what Margaret means is that we have to teach the younger generation that there is more than just the tissue which you see. There's also symbionts. But I think it's equally important these days that we also do not forget this type of teaching what you are now engaged in, and that we have a good and a sound knowledge of the hosts before we, because it goes together. When we want to understand complex processes, when we want to understand disease, we have to understand both, and we have to teach the students.
This is exactly the reason why I created this new master program, Insect Biotechnology. And we have 2 modules, Insect Entomology 1 and 2. In 1, we go through the entire systematics. So each day is a Coleoptera, Diptera, systematics and taxonomy of insects. And 1 module is just focusing on everything, insect development, growth, hormones, to have a basis, to really have a template. And from there, then you can go to the biotechnology and expand to other techniques. That was exactly the reason.
Yeah, excellent. Now coming back to what you told us about your personal history and getting into the field, but now with the view of a new biology. I mean, I guess when you go back in your notebooks and you see your butterflies or your beetles, you certainly you will recognize them again, and maybe you do not find them anymore because they are extinct in the meantime. However, at that time, you probably had no idea how fascinating, complex, multi-organismic these insects really are. And it needed new technology to get into that completely new, I call it sometimes holistic, meta-organismic view. And the question would be, how important is technology in this new field? And you are heading a super technological infrastructure, buildings, companies. How important is the technological aspect in this field?
Without technology, with all of these new things, we cannot do the research that we are now presenting. Keep in mind, we know each other for decades. How I started with Galeria was very biochemical from, let's say, from the Stone Age. So, and over the years, there are new and new technologies coming. We switch to epigenetics, genomics, and this is essential to get a full picture. So that is the reason you need expertise in different fields, bring this together, and then to advance this. That is the key question. And also, I'm saying you should be a visionary. You should really have. I will make an example now. Everybody is aware about the insect decline. I contributed to this white paper, which has resulted in this Artenschutzprogramm. And the government is now interested to spend a lot of money, 180 million euros. And the vast majority of colleagues request funding for monitoring programs. So do I look like somebody who's monitoring? So I try to convince people that it's better to go to the future. We are developing here new technologies. How can you restore biodiversity? How can you resettle insects that have become instincts? For example, flower strips that are made, they could be nice, but they're also cosmetics. If they only produce some, let's say, stuff for the pollinators. So we have made a list of all these plants that are, let's say, sold for the seed, where the seed is sold, and then we have looked how many Lepidopteran species are hosting each of this. And then we make a new composition just to provide also food for insects that have gone. So we go here with the insect farming strategy. So our is combined here. So we have new aspects. How can we, let's say, restoring and resettlement of insect biodiversity? This is one of our research fields also. So, and if you are not going ahead and to say something, everybody will just go to monitoring. We are aware that the insects are declining. And I'm not always everybody's darling. You know, most of the people say it's the agriculture. But I disagree that it is a factor, clearly, but we have others. And I will show you how that even simple questions cannot be addressed. For example, if everybody knows that from your windows, from the cars, that it collects dead insects. If you see a train, it's full of insects. But nobody can tell me how much insect is there. Is it kilogram per day? Is it a ton per day? You have 47 million cars, 10,000 trains. Exactly, moving, let's say, 24 hours, and they kill in the sum more biomass maybe than all pesticides together, because we do not know. Nobody can tell me how big is the biomass of insects in a field or in the forest. There's absolutely nothing. And I try to bring out these questions so that simple questions nobody can ask you. Try to find somebody who can say in 1 hectare of, let's say, oak forest, how many insects are there? Is it a ton, a kilogram? We have really no idea how it is. And the biomass I like these Krefelder study very much, which was not only because they were the first and attracted money, but they were also looking for the right parameter, the biomass, not species diversity. And everybody is now just making red lists and so on. I think this is why you have to inspire people to go into the direction and then to develop strategies how to protect or to maintain biodiversity.
Yeah. So just continuing there, declining insects means, of course, not only the insects, but also the symbionts, the microbes in them, and the symbiotic microbiome. And that fact, of course, stimulates to think about can we A, save that, and B, can we even restore that? And we know that part of your work was enabled or stimulated at least by a huge kind of a biobank from a company, strain collection, which is probably a gold mine for discovering what could microbes, bacteria do. Isn't that also something, when we save it, would that be something where we can restore then kind of diversity and also give it back to the host and then make the host again surviving in a otherwise hostile environment?
Yes, at least you can use the design in ocular. And the advantage of this industrial strain collection, it's official, it's no secret, it's from Sanofi. This is a mixture of different strain collection. It was from the former Höchst, Agrevo, so there are many, so that is the reason why it is so big. And for us, the advantage is it's Nagoya conform because everything has been collected for Nagoya. You can never establish a similar collection now again because it originates worldwide. For example, all the samples that Elf Aquitaine has collected in deep sea drilling, and so all the microbes isolated from there are there. So we have a worldwide picture. We know exactly where all of these microbes originate, and these soil collections decades old. So this can really harbor also, let's say, not only for natural product research, also species that have become insects and to re-inoculate it. That is one of the options that we can provide there.
Yeah. Considering your enormous scope of the work, actually in the insect field, I know only one more person, and he lives in Japan, which has a similar broad scope of organisms and thinking. And so that leads to somebody who is more reductionistic oriented, of course, to the question, can we afford and is it possible that we understand in depth the biology, the physiology, the adaptability of so many different species, or do we need models? Or is any organism in principle a model? Do you consider your organisms, your study objects as models, or is any insect for you actually a potential model?
Yes, that's you know me, as I told you, for decades. When I started, why did I start with Galeria? At that time, when we started, because it was cheap and it was used as a model host for insect pathogens. But you see what has now this is a model for everything. And I started, or the boring beetle, it has also become a model for evolutionary ecology and so on. All of these, or Harmonia has now become a model to study the role of invasion biology, which factors promote invasion biology. I have just sometimes I'm doing also fun research for myself. It's just not applied. For example, I was inspired as a child by the map butterfly, you know, which has a different morphs in spring and in summer. So by hobby, by chance, we compared the role of epigenetics and so on. And now I saw it and I wrote a, let's say, a review just introducing this as a model to study polymorphism. Seasonal polymorphism. So most of the insects that we have started were not models when I started, but they have become models when you publish a number of papers, and others are focusing on the same insect species. So principally, every insect can become a model, but the reasons why they are favored, some models become highly favored, some others not, are different. It depends on the purpose. Have you ever thought, or could you consider that 20 years ago or 30 years ago that Galeria will become such a let's say, prospering model insect used in so many cases?
Yeah, this is of course supported, as we said before, by the development of new technologies, and so that allows them a wonderful deep insight. The same with Hydra. I mean, whoever had thought about what these organisms can tell us all about meta-organisms.
You have made Hydra one of the favorite models for these as a basic organism. So it wasn't when you started, but it has become after decades.
Yeah, exactly. So I just had a paper with Margaret where we make the point that today, because of technology, actually any organism which you, with your deep zoological, biological knowledge, consider as interesting for some reason, can be made a model. And I think your case, and maybe also our case, is a good example. And Margaret works on the Hawaiian squid, which is a very obscure night-active animal, but she made it as a world-known model system for symbiosis. So you are famous, leading, honored, highly recognized as a biotechnologist, yellow biotechnology, insect biotechnology. And we learn so much. I also learned so much right now after your talk, or during your talk, what microbes can do. You're very much focused, of course, and for good reason, on what microbes can do for the insects, and they allow them to eat on things or live on plants or wherever where they normally without the microbes couldn't live. In the human microbiome field, it's of course the same thinking, to understand what do the microbes do, but then at the end use the microbes to improving health. And that again goes back 100 years, and it goes back to Metchikov and to his famous kefir or yogurt stories, if they are true at all. My question would be, is that a terminology and an interest in the insect field considering probiotics? We always say this is, of course, the challenge of the 21st century, also particularly in the human microbiome, but at the moment we are not yet there. All the probiotics what we get, my colleagues tell me, actually we do not know what they are really doing. You are now so deep in the genome, in the proteome, in the peptidome, in the epigenetic of the microbes. Do you think that kind of a probiotic which is understood and actually extracted as a formula, is there a future for that?
Yes. One of the most rapidly expanding fields in insect biotechnology is the insect farming. You know that it's worldwide boom. It has become a billion market. And insect farms they are considered now or they are growing for alternative protein generation, because we can only afford to eat so much meat because of the deforestation in South America for soybean. So we need alternatives, sustainable protein production system, and here insects are, let's say, number one at the moment regarding the development. But if you have a mass rearing and you produce tons, millions of tons, and you have so many insects with a short cycle, you can expect that sooner or later you have also to cope with pathogens. Insect farming has the advantage it has a very good ecological footprint. You need a low amount of water, only in the areas where there's no greenhouse emissions. And one of the beneficial aspects that we want to maintain is that we don't want to use antibiotics, even if you culture tons of insects. And we are developing for insect farms concepts and strategies how to prevent the growth of antibiotics, and one of these is based on probiotics. We were the first reporting and describing probiotics for insects, and we are developing for big farms like Insect in France, for example, was inspired by Yellow Biotechnology. They have now acquired funding about 260 million euro for a new farm only based on mealworms, and they also interested to have probiotics to make the mealworm more robust. The technologies we are developing see how can you prevent inbreeding depression, what kind of hygiene standards has to be maintained in the farm, and this will become, let's say, worldwide standards. And therefore, the probiotics are now booming. There are also approaches to protect bees by feeding of probiotics. It's expanding in insects now.
You made a key word, and that's antibiotics. One of the major concerns also in our communities, of course, that we have a still increasing uptake of antibiotics, and particularly humans, but also in agricultural animals, in animal farming. And that is, at least according to Martin Blaser, and he got very much supported by many different studies, one of the major causes of microbial disappearance, and particularly the diversity. We are looking, and we touched that when you talked about your Autobahn experiment and the glass shields on the cars. We are realizing a changed world, and we are realizing that in this changed modern world, we have an increased number of lifestyle diseases, and the lifestyle diseases are correlated with chronic inflammatory diseases. There are chronic inflammatory diseases, and there seems to be in nearly all of them dysbiosis, particularly in the gut, but also the skin, etc. How can you follow that in your research, that in the last, let's say, 50 years, there's a dramatic change for the insects, which makes them very difficult to survive, or developing diseases, or just disappearing? And can you correlate that to a change in the symbiotic microbiome?
There might be some published data, but I'm not aware of this, whether we have a change in the microbiome over time. we are focusing on other instances. That is what I told you today is I would like to see whether the pathogen prevalence has changed over time. And therefore we have access to the Senckenberg, to the species collection, so we can determine even in dead insects whether you can determine DNA of viruses or bacteria. And then we can see whether over time pathogen prevalence increased. But this is a very demanding project. But this is the only one I know, and this is what we are working on. I'm not sure whether the microbiota has changed, but I would like to give you other examples where we are working inside. The black soldier fly is very nice. It's, according to my opinion, aside from the bee, the most important economic insect because it's worldwide used. And we are assisting companies worldwide to become bigger farms. And what I can say is that there's really a rapid adaptation. They are not native to Indonesia, for example. We are now working for a company that would like to have the palm oil fruits, empty fruit bunches. You know, there's billions of tons. And you press it, you cook it, then you have the empty fruit bunches that are burned. It's a lot of organic material. And the idea was to use this as a diet for black soldier fly. But they cannot feed it, so we are developing a fermentation process. But now we have discovered a population in Indonesia which cannot, let's say, they must, let's say, only the maximum 100 years, but they have completely changed the microbiome and adapted to the digestion of palm oil fruits. And now this local population is now used for local process to establish an insect farm. All other strains failed there. And here you can see that even in a relative short time, these black soldier fly has adapted to a new niche, and this is, we can attribute it to the microbiome.
It's fascinating. It also tells you it always needs an excellent observer, naturalist, entomologist who actually knows about the abundance and the presence of this type of species in certain parts of the world. We have talked a lot about already about future developments, and you are leading in developing new novel technologies and using more and more the different species. What is a major open question for you in your field?
There are so many fields that I have to prioritize first. So my interest is now, one of the biggest superior goals of my life is to leave a better world than those I have, let's say, that I perceived. So I try to improve on many balances. One is the teaching activities. It means to invest in offspring. In terms of biodiversity, I would like to have more time to invest in these new strategies, how to restore biodiversity, particularly in agricultural landscapes. We have become part of a consortium that has applied for a EU project, so many countries together developing these new strategies. That's what I really impressive. So because this is what we could give back to nature, I would like to develop further technologies to make everything sustainable in a way. So we have here the Agricultural University. So that is to development of alternatives to chemical pesticides is something which is rapidly booming. I think we are the leading group in Germany developing these new technologies for plant protection. For example, the RNAE approach, you know, this RNAE silencing. And we have a long-lasting collaboration had with Dow, and we engineered several plants which are resistant against the corn rootworm. But these engineered plants are GMOs, so there's no market in Germany and in Europe. But now we are going back to, you can also make it sprayable. And we are now developing sprayable approaches. It means formulations. And we are here, we have started with the first field trials. And when I see how strong I have, the strongest support in Germany I have are the farmers. They said they push the governments to give us more money because we are the only one developing alternatives to these pesticides which have been banned. And this is something where we got funding. So the biological control options that I report today is only one part, a small part of these entire activities. And this is something, if you see a product on the market and you see you can help people, you know, working for decades with antimicrobial peptides, but now we are in a phase where we design products that are coming on. I have a company, you know, the wound maggots. I'm always reporting this in my previous talks. We have isolated, we determined 47 different antimicrobial peptides, 23 could be produced. And with Sanofi, we had a partner who invested a lot. So we selected 100 antimicrobial peptides from insects and make an industrial profiling. It means not only testing whether they display some activity against gram-negative, we tested against all the escape strains, all multi-drug resistant strains, and also clinical isolates. Then you have to figure out whether the activity is maintained when you have physiological conditions. Then you have to see whether you have side effects, whether development resistance. So there's a catalog of rigorous industrial criteria, and only 2 antimicrobial peptides survived. And they are now further developed. On one hand, we make them particle-based to see when whether you have an inhalation in the lung, because this is ectopic application. So we are now, along with another Fraunhofer Institute, in the mouse model. We have a company that is producing these peptides now for skins, as a wound care skins. So we hope that we can sooner or later have a product on the market which helps. And this is what for the future, that we see something we have contributed to research resulting in a product or a service which benefits humans. This is my ultimate goal. Because Hirsch factor or nothing else, on your gravestone, you will not have something like this. And what you left is a better world. That is what we would like to develop here. And this is the common spirit in my group, that I have a lot of people here contributing to this idea to make it better, to make something which is better, and without us, you would miss it. And this is not necessarily papers and so on.
Yes, that's actually an excellent point, Andreas. And we have to come to an end, but the last question actually goes directly from there. What do you think it needs, or what you would like to tell the junior generation, an undergraduate who sees something what you're doing and he or she is questioning, is that something I can do? What does it need to become a successful scientist and to do well in this field, from your point of view?
I think you should be, first of all, inspired. Fascination of insect is that what drives me. Then you should be highly motivated, dedicated, and committed. It means you have to work a lot. I'm not excellent because of the paper. It's a lot of work that we are doing. You need to be passionate. You have really to show that you have passion for your field. Then sooner or later, everything comes from yourself. This is something you cannot educate. If you cannot, let's say, put a light in somebody, and you need these eyes that are really motivated and having fun. These are the good people. And I try to install this in the students that we have. And I'm highly, I would say, also successful with my daughter. She's 8. We have a lockdown. Every lockdown she has a new aquarium. Now she has a terrarium with snakes. So she's now, she is all the time. She starts in the same direction, at least. I'm happy to see this. She's now breeding different fish. So she started with guppies, and now she has she likes it just to breed them.
How wonderful! I mean, I couldn't agree more, Andreas. And I think it needs shining eyes, and so intrinsically driven, and also that means that you work a lot, but it's actually also most of the time kind of fun, and that is which makes you so successful. And if you can now transfer that to the next generation in your daughter, but also to hundreds of undergraduates in the university and in your institute, that's of course just wonderful. I really like to thank you for sharing personal views with us and giving a fascinating insight in not only your science, but also in your thinking behind. And wish you all the success for the future. And we are very much hoping that we can meet each other in person soon. Thank you so much for joining us and for this wonderful chat.
I thank you. I appreciate it, really. I hope that we can really meet soon. maybe again like a meeting like in Crete or something. I hope that we have some time together and to enjoy also then. Thank you, Thomas.